Namaste and welcome back to Typical Monk. Every living cell needs energy, and today we see how it is made. We will follow one molecule of glucose as it is broken down step by step to release ATP through glycolysis, fermentation, the Krebs cycle, and the electron transport chain. And we will drill it the neat way too, one idea at a time. Here is today's path. 5 stops. First, glycolysis, where glucose is split in the cytoplasm. Second, fermentation, that is respiration without oxygen. Third, the Krebs cycle inside the mitochondrion. Fourth, the electron transport chain and how much ATP we finally gain. And fifth, NEET drills, that is assertion, match, and statement. Let's begin with the very first step, glycolysis. Glycolysis, also called the EMP pathway, happens in the cytoplasm, and it is the one step common to all respiration, whether oxygen is present or not. Here, one molecule of glucose, which has 6 carbons, is broken into 2 molecules of pyruvate, each with 3 carbons. The net gain is 2 ATP by substrate-level phosphorylation, plus 2 molecules of NAD. And pyruvate is the crossroad. Its fate decides which pathway comes next. Now, what if there is no oxygen? Without oxygen, pyruvate is only partially broken down, and this incomplete pathway is called fermentation. In yeast, pyruvate is turned into ethanol and carbon dioxide. This is alcoholic fermentation. In our own muscles and in lactic acid bacteria, pyruvate is turned into lactic acid instead. Very little energy is released, a net of just 2 ATP. The NAD is reused so that glycolysis can keep running. Now bring oxygen back and we move into the mitochondrion. With oxygen present, pyruvate enters the mitochondrial matrix. In the link reaction, pyruvate is converted to acetyl-CoA, releasing 1 carbon dioxide and 1 NAD. Acetyl-CoA, which has 2 carbons, joins oxaloacetate, which has 4 carbons, to form citrate with 6 carbons, and the cycle turns. Each single turn yields 3 NAD, 1 Fa2, and 1 ATP, and releases 2 molecules of carbon dioxide. Now the final and biggest energy harvest. The NAD and Fa2 made earlier carry their electrons to the chain, sitting on the inner mitochondrial membrane. The electrons flow through the complexes, and oxygen acts as the final electron acceptor, joining with them to form water. The proton gradient, built across the membrane, drives the enzyme ATP synthase, and this making of ATP is called oxidative phosphorylation or chemiosmosis. As a rough count, each NAD gives about 3 ATP, and each Fa2 gives about 2 ATP. Let's add up the balance sheet. The complete aerobic breakdown of 1 glucose gives a net of about 36 ATP, as stated in NCERT. Fermentation gives only 2 ATP, so oxygen makes respiration far more efficient. The respiratory quotient is the carbon dioxide released divided by the oxygen used. It is 1 for carbohydrates, below 1 for fats, and above 1 for organic acids. And remember, the respiratory pathway is amphibolic. It both breaks down and builds up molecules. Now, the high-yield pointers, the facts that NEET turns into questions. 1. Glycolysis happens in the cytoplasm. Glucose becomes 2 pyruvate. The net gain is 2 ATP and 2 NAD. 2. The Krebs cycle runs in the mitochondrial matrix. Each turn gives 3 NAD, 1 Fa2, 1 ATP, and 2 carbon dioxide. 3. The electron transport chain is on the inner mitochondrial membrane, and oxygen is the final electron acceptor. 4. Each NAD gives about 3 ATP and each Fa2 about 2 ATP, for a net about 36 ATP per glucose. 5. Respiratory quotient is 1 for carbohydrate, below 1 for fat, above 1 for organic acid, and fermentation gives a net of 2 ATP. Now the first drill, assertion and reason. Each time, ask, is it true? Is R true? And does R explain A? First, A, fermentation releases far less energy than aerobic respiration. R in fermentation glucose is only partially oxidized, both are true, and R correctly explains A, so the answer is second. The Krebs cycle can run in the complete absence of oxygen. R. The Krebs cycle enzymes are located in the mitochondrial matrix. Here A is false, because without oxygen the cycle soon stops, but A is true. So the answer is D. Third, ATP synthase sits on the inner mitochondrial membrane. R. The proton gradient across this membrane drives ATP synthesis. Both are true, and R explains A, so the answer is A. Now, the second drill, match the column. Make the correct pairing. Glycolysis, the cytoplasm. The Krebs cycle, the mitochondrial matrix. The electron transport chain, the inner mitochondrial membrane. And the final electron acceptor, oxygen. Now the third drill. How many of these statements are correct? Check them one by one. First, glycolysis produces a net of 2 ATP. This is correct. Second, the Krebs cycle occurs in the cytoplasm. This is wrong. It occurs in the mitochondrial matrix. Third, oxygen is the final electron acceptor in aerobic respiration. This is correct. So the correct statements, 2, that is, the first and the third. Now let's clear the mistakes students fall for. Myth: glycolysis needs oxygen to run. Truth: glycolysis is anaerobic. It runs with or without oxygen, right there in the cytoplasm. Myth: fermentation and aerobic respiration release the same energy. Truth: fermentation gives only 2 ATP, while aerobic respiration gives about 36. Now test yourself. Answer each one before it is revealed. Answer first. That is how you build strong NEET recall. Question 1. Where does glycolysis occur? The cytoplasm. Question 2. The net ATP from glycolysis? 2. Question 3. Where is the Krebs cycle? The mitochondrial matrix. Question 4. The final electron acceptor in the electron transport chain? Oxygen. Question 5. The respiratory quotient of carbohydrates? 1. Let's gather it all in one breath. Glycolysis is in the cytoplasm. Glucose becomes 2-pyruvate. The net gain is 2 ATP and 2 NAD. With no oxygen, we get fermentation, ethanol or lactic acid, and only 2 ATP. With oxygen, the link reaction and the Krebs cycle run in the matrix, releasing carbon dioxide, NAD, and FAD2. The electron transport chain sits on the inner membrane. Oxygen is the final acceptor, and ATP synthase makes the ATP. The net gain is about 36 ATP per glucose. The respiratory quotient tells us the substrate, and the pathway is amphibolic. In the next class, plant growth and development, including growth phases and plant hormones. See you there. You have followed glucose all the way from the cytoplasm to ATP, the fuel of every living cell. Be proud of that. One idea at a time, with a calm mind, and you will crack NEET. This is Typical Monk. Every class, always free.